Okay, thanks for joining everyone, and welcome once again to the Coffee Science Seminar. Raisa, can you go to the next slide? Thank you. So you're here for a presentation by Dr. Raisa Rudge, who's going to be talking about future foods using ancient fermentation techniques. So stay tuned. I've got some housekeeping to do first, but we'll get on with the seminar in just a moment. Just next slide, please, Raisa. Sorry, mate. So my name is Heather Smyth, as you well know, and I'm Deputy Director for Research at QAAFI, but I'm also a research fellow working in sensory science and flavour chemistry. So I'm going to be chairing and facilitating the science seminar with Raisa this afternoon, sorry, 3 minutes this afternoon. Next slide, please. Thank you. First, I'd just like to acknowledge the traditional owners and their custodianship on the lands on which we meet. And for me, that is the Yagara and the Turrbal people. And I pay my respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We also recognise their valuable contributions to Australia and global society, and also to coffee and the rich research that we do here involving many of our Indigenous partners. The seminar today is scheduled from 12 till 1, and we won't go over that time. After the presentation, we'll hold a time for questions for Raisa to answer. I'm just going to ask you to please type your questions in the Q&A tab. That allows us to see them at the panel end, and I'll deal with those at the end of the session. But as a question comes to mind, feel free, please, to pop it in the Q&A tab. Don't use the chat function. So not the chat function, Q&A tab. Thank you. All right, so now on to introducing our speaker for today. So Dr. Raisa Rudge has a background in chemistry and chemical engineering from Leiden University and Delft University of Technology in the Netherlands. She completed her PhD in food physics at Wageningen University in 2021 on the subject of soft matter friction and lubrication. In particular, her work involved understanding how tribological measurements can be applied to evaluate a range of different food systems. In 2020, Raisa came to Australia and joined the UQ Chemical Engineering Group working in food with Jason Stokes as part of her PhD project. And after completing her PhD in 2021, Raisa began her postdoctoral research project on plant-based meats with optimized textural properties and limited ingredients using fermentation on an ARC linkage project. She was working with me in the Centre for Nutrition and Food Science at QAAFI. Now, almost 3 years as an ARC postdoc, Raisa is about to transition onto some pharma projects and food and beverage accelerator projects, working with clients who are developing foods with precision fermentation. And her presentation, I've already mentioned that today, but the name of that presentation today, it's not on the slide, but Raisa, I'll let you get started. Over to you.
Thank you, Heather, for that introduction. And I too like to start by acknowledging the traditional owners of the lands on which we meet today, and I pay my respects to their ancestors and descendants. So Heather has already given a very nice introduction to transition us into the actual presentation today, entitled Future Foods Using Ancient Fermentation Techniques, and I'll be discussing fermentation, sensory, and beyond. So we have a few different things to cover today in the next hour or so, and this is, well, I've been researching for a while, but of course I'm a relatively young researcher, and I realized today that this is actually the longest presentation of this kind that I've ever given. And yeah, I'm just very excited to share an overview of what I've been working on, of some of what I've been working on in the past 3 years, and I also look forward to discussing in the Q&A section. So I'll keep this short, as some of this was already mentioned and was already in the invitation as well. So I do have a background in chemistry, chemical engineering, and as you'll see throughout the presentation, any chance I get to talk about molecules or show any molecular interactions, I will definitely take, because I do think that's the fundamental, the building blocks of all of the food and also agricultural research we do. In 2014, I got a taste of food science at Wageningen University during my master's as a short exchange, and I soon realized that I liked connecting the molecular interaction, so the chemistry, to something a bit more tangible, something you can actually discuss at birthday parties, such as food. So during these electives, I got to work with ice cream, with wine, with chocolate, and from then on, it was actually really hard to turn back to basic chemistry. I was definitely sold on food. And I also took a course in food history, which during that course, I actually learned a little bit about ancient fermentation as well, which ties into this presentation. My PhD at Wageningen University, as Heather just mentioned, and I got to do a placement all the way on the other side of the world. And I'm still here at QAAFI, carrying out my postdoc research. So where my research interests lie, as the previous slide actually already showcased, I have quite a broad interest, but I do think there's some synergy between those interests. And so I'm interested in structural properties and how they relate to sensory properties and performance, where food processing comes into these different pillars, and of course also the characterization, and at the moment specifically characterization methods that are relevant to sensory evaluation, so that help us better understand how physical measurement, physical characterization ties into what the sensory panel, or what us as consumers, will be actually experience. So that's enough about me today. This is a brief summary of everything I will be discussing today. Most of this probably does not look familiar to you, which is great, because that gives me enough room to introduce these topics to you. But before we get into all of these excited foods, let's just start a little bit closer to home. So it's 12:10. Most of us have had breakfast. Some of you may be having lunch, which means that probably you've already encountered a few fermented foods. Yogurt, fermented milk, cheese, also from milk, dairy products, bread, fermented grains, coffee beans, actually also fermented, and wine, of course, fermented grapes. So fermented foods are not new. They're not unknown to us at all, which is a point I wanted to make before getting into the newer side of the potentially newer side of fermentation for some of us. So the foods you see here in the background, although they might be new to you or unknown to you, they are actually consumed by about 75 million people daily. They cost average $3 per kilo. They are naturally healthy, which depends on what your definition of healthy is, of course, but I think we'll probably be able to agree by the end of the presentation that they are naturally what we generally accept as healthy. And importantly, they are made via a very simple production process. And I'll be telling you much more about this soon. So a little bit of food history. Again, fermentation, it's not new. I have a bit of a timeline here, which starts at 10,000 BCE, where in ancient China, vessels were found that were used back then for fermenting, probably something similar to beer, wine, or mead. In Mesopotamia, one of the early ancient civilizations, maps were found indicating the location of beer, images of people drinking beer in Egypt, and wine production also in those early days. So these are all images that tell us that fermentation was actually performed very early on. So what actually is fermentation? So fermentation can be described in many ways. The description that is relevant today is the transformation of food by means of microorganisms such as fungi and bacteria. Essentially, you start with something very simple, which I've showcased by just showing a simple oval. And in this food system, there is some sugar, protein, fats, carbohydrates, which the microorganisms use and actually transform. And what comes out of that transformation depends on the time, temperature, humidity, the type of microorganism used, etc. And this might lead to fermented foods that look very different from where we started. And these foods often contain soluble peptides, fats, very exciting aroma and flavor compounds, but also things that weren't initially present, such as mycelium or even biopolymers that are created during fermentation. So here I just want to showcase what the before and after looks like. So on top, you can see some of the legumes that I used: soybeans, African locust beans, faba beans. And if you look at the bottom of the chart, you actually can barely recognize these foods after fermentation. And if you just have a quick look, I'm pointing out ukpa over here, which is an African product. It's boiled, sliced, fermented, and then ends up with something that looks completely different to where we started. So why in 2025 are we still interested in these ancient fermented legumes? So one of the reasons, and a reason that's particularly important for this project, is because of their potential in plant-based foods. Of course, legumes are from plants, are plant-based foods themselves. So something we can ask ourselves is, what potential do these foods have as applications in plant-based systems? So again, this might be a little bit closer to home than some of the ancient fermented foods I've shown. We have, so these are just commercially available plant-based meats, which I'm sure you may or may not have seen these or similar products. So I'd like to share a little bit about how these plant-based meat alternatives, or PBMA, are actually made. Similar to some of the fermented foods I've shown, they start out very simple as a humble soybean. That's then processed into a protein concentrate, which means that the proteins are extracted from the beans. That's then further purified to have a protein isolate, so high quantity of protein. And then further processing gives something called texturized vegetable protein, or TVP, which I'll be mentioning a few times throughout the presentation. So how do we actually get here? It involves things like chemical processing, but also high temperatures, high pressure. And to actually get from texturized vegetable protein, TVP, to a plant-based burger, as you may have seen it in the supermarket, requires the addition of some additives, which essentially, very simply put, glues the texturized vegetable protein granules back together to resemble a burger, or maybe even a nugget or other food products that may be on the market. So this is, of course, I think it's very impressive that we are able to get from a soybean all the way to something that resembles a meat burger. However, something we do need to think about is that although we get a chewy, firm texture, which may remind us of meat, it does require excessive processing. It reduces the functionality of the protein, and it sometimes comes with poor digestibility as well. So some of these TVP products have texturizers, flavoring, and coloring added, because we do want something that resembles animal-based meat. And so it's quite the process to get to where we want to be, which is a reason to look into fermented legumes as plant-based meat alternatives. So some of the research aims of the projects that I've been working on include understanding how legumes obtain meat-like properties without the excessive processing and with limited additives. So some of the questions we asked are, how is fermentation affecting the protein content? How is fermentation affecting the protein functionality? You can think of solubility, gelling, etc. But then also, can we translate these findings, so what we learned from these fermented foods, how can we translate this into Western foods, so foods that people are familiar with, such as burgers? We may be able to inform manufacturers on more sustainable processes to make plant-based meat alternatives, and also normalize new flavors and textures from fermented foods. Because the reality is some of these fermented foods do have very interesting and pungent flavors, but I do think that if you can eat or get used to something like Vegemite, then some of these fermented foods should be able to fit that palate as well. So some of what I'm going to share now is from a recently published review. Review was published in Food and Function, and it's called Insights of Traditional Fermented Legumes Towards the Innovation of Modern Plant-Based Meat. So how can we learn from ancient fermentation techniques for a new generation of plant-based meat? So I'll now introduce some of the foods I've shown earlier. So we have tempeh, natto, dawa dawa, kinema, and silio. And I hope that you've heard of at least one of these. I think tempeh is one of the most popular ones, and also probably the only one you might be able to find at your local supermarket in Australia. So tempeh is originally from Indonesia, natto from Japan, dawa dawa is commonly consumed in West Africa, kinema in Nepal, and silio is consumed in East Africa. And they're all made from their own legume types, but also different bacteria or fungal microorganisms. So what I can tell you about these foods already is that they have a high protein content, from 20 to 50%, and the protein content actually depends on the fermentation conditions. As a comparison, a beef burger has about 15% of protein. The textural properties are, as you can tell from the pictures, they're very broad, they vary a lot, but some of them can be described as chewy and juicy. They have all these interesting flavor compounds, but umami is one that most of these foods have in common, which is also a common flavor attribute of meat. As I mentioned, they're made of beans in a simple conversion process, which makes them affordable and sustainable. And a lot of these foods are naturally rich in iron and B12, similar to meat. So I'd now like to dive a little bit deeper and introduce some of these fermented legumes in a bit more detail. In particular, tempeh, natto, and dawa dawa. Tempeh can be described as a firm cake with nutty and umami flavors. Natto can be described as soft, sticky beans with umami, maybe like a strong parmesan or cheesy flavor. If you look closely at the natto, you can see some of the strings, and that stringy texture is actually quite noticeable as well when consuming the beans. And dawa dawa is a bit of an interesting one. It's very lumpy. It has a paste-like texture, but the flavor is very potent. It has a strong umami, gamey flavor. Dawa dawa isn't actually eaten as a meat replacer or eaten as a high-protein food, but because it's so intense in flavor, it's used as seasoning. So it's actually sprinkled on meals or added to soups and stews. So to obtain tempeh, a 28-hour fermentation step is required. So that's how simple it is. You add the rhizopus fungal strains, and you go from a soybean, as we know it, to a high-protein cake that has a protein content up to 50%. In this case, when I say up to 50%, that means that I was able to find sources in literature where they optimize the fermentation process by using different legume cultivars, by maybe extending the fermentation process, fermentation time, and just tweaking the conditions to achieve that high protein content. So that means that the tempeh you buy at the supermarket will likely have a slightly lower content, which I think is worth mentioning, but the potential is there. During fermentation, the carbohydrate content is decreased. Iron and B12 are generated. Mycelium is formed, which is the white encasing you see in the image, and umami flavor molecules are created. This is a microscopy image of the mycelium that's created around the tempeh system. Next, we have natto, which, as I mentioned, is a product from Japan. In this case, we have a bacterial fermentation. And the fermentation leads to up to 45% soluble protein. And the reason why I highlight soluble protein is because a lot of plant-based proteins are actually poorly soluble in water. And after fermentation, these proteins are quite well soluble in water, or there's an enhanced protein solubility. The iron absorption is enhanced, vitamin K2 is produced, mucus layer is produced, and umami flavor molecules are also created in this case. Next, we have dawa dawa, as I mentioned, from Western Africa. This requires a 36-hour fermentation, again, a bacterial fermentation. And in this case, it's an alkaline fermentation, which is interesting because most of the other foods I've mentioned actually undergo an acidic fermentation. In this case, we arrive to up to 40% soluble protein. Iron and zinc bioavailability is approved. There's a sticky layer formed again, and intense, very intense. I haven't had the product myself, but from what I've read in literature, it's a very intense umami and roasted, maybe even coffee-like flavor compounds. And a lot of work on dawa dawa has actually been on reducing the intensity of the flavor and aroma, which already tells us that it's quite intense. And if there would be an interest in using this as a plant-based meat ingredient, that's something that would probably have to be optimized. So I'd also like to show the nutritional value compared to burgers. So just again, what I just showed previously are the highest values I was able to find in literature. What I'm going to show now are the average values that the USDA presents based on what's commercially available. So in this graph, I have soybeans, tempeh, natto, lean beef, a beef burger, and an impossible burger, which is a plant-based burger. I'm going to skip over the water, but if we look at the protein content, we can already see that it's all in the same range. Actually, impossible burger scores and beef burgers score lower in protein than some of the fermented foods. If you look at fat, a beef burger is quite high in fat, a lean beef burger is lower in fat, but the tempeh and natto has a lower fat content than these beef burgers. Carbohydrates is a challenging one, as beef ingredients generally do not have carbohydrates, but we can also see that some of the fermented products are also quite low in carbohydrates, which is what I wanted to showcase here. And I'll briefly go over this. These are the flavor molecules in fermented legumes. So what you can see here is some of the flavor molecules that are found in fermented foods. So one of them is glutamic acid, or MSG, which contributes to a umami flavor and is also present in animal tissue. I'll show some nucleotides as well, which also contribute to umami flavors. And these are also found in beef, chicken, pork, and duck. And then some fatty acids, compounds which are found in fermented products and also in meat fat, which explains why some of the fermented foods have a similar umami and maybe even sweet flavor that is associated with some meat products. Again, highlighting the potential of these foods as meat replaces. So the textural properties, I already mentioned some of this, but fermentation by bacteria or fungus gives a completely different texture and even color and flavor than soybeans. And what I can share here is that, which is also one of the things we talk about a lot in the review, is that the water holding capacity is improved, the cooking loss is improved, firmness, and also the protein solubility, or the solubility in general, which are some of the advantages of fermenting legumes. And this can all be modified based on fermentation time, legume selected, the microorganisms selected or combined, fermentation temperature and fermentation humidity, etc., but also the post-fermentation treatment. So think of cooking time, cooking methods, additional ingredients that are added. So I hope I've convinced you that there is a lot of research potential for these fermented legume ingredients. And some of the things we can ask ourselves is what flavor and texture properties are generated during fermentation, but also what properties can be obtained post-fermentation, which opens up another realm of possibilities. So in the next section, I'd like to show you some of our experimental work, where we show how we can use fermentation to obtain less processed PBMA. So for this research project, we actually fermented tempeh in-house. So we carried out our own fermentation, and we started with soybeans. And soybeans were cooked and dried, combined with fungal spores, and incubated for 28 to 30 hours. And this is what our fermented product looked like. This is one of the first, or for me, probably the actual first fermentation I carried out in a lab. And I was actually very proud to see that the mycelium actually grew as expected. And we actually eat these products as well. And they actually, yeah, they tasted quite good. I was quite happy with that when we first achieved that. And now we get back into the post-fermentation properties. So how do binder properties affect tempeh texture? How can we use binders to control the final texture? So for this, we use methylcellulose. Methylcellulose is an ingredient that's commonly used in plant-based foods as a strong binder. Egg white, which is also commonly used in animal-based burgers for moisture retention, gelling. Gluten, which is known for its viscoelastic properties, and we use vegan egg, which consists of, it's a commercial product, it consists of starch, proteins, polysaccharides, and those combined ingredients are expected to contribute to moisture retention and binding. So we use these ingredients to make tempeh patties. I'm going to show you a few of the images, and based on the images, you can probably already tell that they differ in texture. So this is the plain tempeh patty, which essentially means we ground the beans and just compress them using a burger press. This is the burger with 5% gluten. Here we use 10% egg white, 5% methylcellulose, and a mixture, and that mixture contained 1% methylcellulose. So just these images already show the different appearances we're able to achieve. We use these tempeh patties for a sensory evaluation. Sensory evaluation is one of the main things we do at our lab, and carrying out a sensory evaluation with our trained panel on these patties was, a very important step in this research project. So we had 14 trained sensory panelists. We carried out 10 sessions, and we used a descriptive profiling method. So I just want to briefly introduce the way we tend to present sensory results. Here I'm showing what we call a PCA plot, and I'll keep it as simple as I can. The red lines show axes that are relevant to the sensory attributes. So for example, roasted over here, this is the axis for roasted, and if a sample, so the purple dots are the results for the samples, if it's very close to this, to the high end of this axis, it means that there's a very high roasted flavor, and it's far away from legume, which means that it has a very low legume flavor. So for the flavor properties, we found during sensory that the plain sample, so with nothing added, just compressed tempeh, has a high flavor intensity, but it's also roasted and bitter. Adding egg shifted the flavors toward more legumy, fermented, and bitter and roasted flavors went down. Vegan egg increased legume flavor, decreased bitter and roasted flavor. Gluten increased fermented and decreased roasted flavors, and methylcellulose increased legume flavor and decreased roasted flavors. Now I have a similar overview for the textural properties. In this case, the textural properties that were of importance were moistness, cohesiveness, crumbliness, lumpiness because of the beans, firmness, and density. Some of our main findings here is that the plain texture is crumbly, lumpy, and moist. Adding egg white had limited effect on the texture. As you can see, these samples are all quite close to each other on our projection on the PCA map. Gluten was able to enhance cohesiveness and decrease crumbliness, while vegan egg enhanced moistness and cohesiveness. And as you can see, all the light blue samples that are samples that contain methylcellulose are all towards that firm, dense side of the PCA plot, as methylcellulose enhances firmness and density, but strongly decreases moistness, as you can see in the PCA plot. So just by using 5, or actually 4 relatively straightforward ingredients that are commonly used in foods, we were able to obtain a range of sensory characteristics based on flavor and texture simply coming from fermented legumes. Next, we carried out our texture profile analysis. As you remember, I mentioned at the start that I'm very interested in connecting characterization methods to sensory properties, which is what we did here. So in this graph, I showed the sensory score in the dark blue bars and the texture profile analysis, which is a compression method, with the white dots you can see here. So texture profile analysis is a very commonly used technique in food science, and a compression, so a force is applied, and based on that, we receive a readout of essentially the hardness or the resistance of the sample measured. And here we see that if you look at the trend of the bars and the trends of the diamonds, we actually see that our sensory results and our measured TPA results very strongly follow the same trend. And we see this for cohesiveness as well. But what I want to show here in terms of how our binders, how the ingredients we added behave, we find that methylcellulose, as we saw for sensory as well, increase hardness and cohesiveness. Gluten increases cohesiveness. Vegan egg, again, increases cohesiveness. Egg has limited effect on texture. If you look at plain and the ingredients with egg, hardly anything happens. Whereas if you look at methylcellulose, adding 5% methylcellulose increases the hardness up to 10%, which is a very large increase considering the small amount of binder that's added. So here I've included the microstructures. These images are obtained using fluorescence microscopy, and I'll show the images from increasing firmness and hardness. So this is the plain sample. As you remember, it had a very low firmness in sensory and a low measured hardness. And if we were to describe this image, we can see a lot of loose cells. There's not a very tightly bound network that's visible. For egg white and gluten, we can see that there's a lot more protein. So green is shown in protein here, and especially in egg white, that protein, of course, comes from the protein content of the egg. And similar for gluten, which also consists of protein. So we can see that there's added protein. However, it does not look like a really strong network or tightly bound cells in this case. And for methylcellulose, which we already know has the highest firmness, you can actually see that where we go from a loose network here, we go to a very tightly bound network, which explains the high cohesiveness, high hardness, and high density. So we have a system with no binder, a system with weak binders, and a system with a very strong binder. So to summarize this section, ancient fermentation techniques were successfully used to obtain high protein patties using limited processing and ingredients. Both flavor and texture were affected by the binders. Methylcellulose increases firmness and decreases moistness. And the TPA results were in agreement with sensory. So we now know that we can make these patties from soybeans with limited processing. But where does that put us? How far along are we? Are we in the right direction for something that could potentially replace meat? What textural properties are required to achieve that? So I did a little deep dive into literature, and I found that a lot of papers mention juiciness and tenderness as main textural attributes in meat. Juiciness, again, mentioned in another paper, and here we find, again, juiciness should be prioritized. High juiciness leads to high overall liking. And this is where we get into a bit of a challenging area, where meat analogs lack of juiciness, elasticity, and firmness, when we actually just learned that juiciness and firmness are important attributes for meat. I want to share with you some results that were obtained by my colleagues within the same project that I'm working on as well. In this project, we looked at a range of commercially available burgers. So what was done here is the animal-based meat products are shown in red, and the plant-based meat products are shown in green. And we see that there's a clear distinction between them. Some samples are further apart than others, but we can actually see that there's still room for growth within this space of plant-based meat. So we've been talking a little bit about juiciness, but what actually is juiciness? Which is another challenge because literature also shows very different descriptions of these attributes, such as the amount of moisture perceived during mastication, intensity of juice in the mouth before swallowing, so that those are already different time points during mastication before swallowing, and the amount of liquid released when chewing the sample. So very different descriptors for the same sensory attribute to juiciness. So there's a lot we can still learn and a lot we still need to understand about juiciness before even attempting to create samples that are juicy. So for this, we asked ourselves two questions in the next project. How does sample formulation and sample preparation influence sensory juiciness? And how to accurately measure juiciness mechanically? Because if we can't agree on a word or on a description for juiciness, maybe we can agree on how to measure juiciness and have a universal method to measure juiciness, which also removes a lot of discussion on what actually is juiciness. So for this, we used model burgers. As the emphasis was on developing the method, understanding juiciness, we resorted to a simple and straightforward model burger system. For this, we used TVP burgers and beef burgers. We varied the oil, water, and methylcellulose content as we hypothesized that changing those ingredients or the ratios between those ingredients would give us a range of juiciness scores. And we also included different methods of methylcellulose addition, which is also something novel that was done in this work. We carried out sensory evaluation and mechanical measurements. So I'll walk you through the experimental design. As I mentioned, we used plant-based and animal-based burgers. We used methylcellulose, which was added as a powder or as a foam, and various ratios of oil and water between 0 and 10%, and also different internal temperatures. So why did we use different internal temperatures? Is because, again, we're trying to adjust the juiciness. And as you can imagine, cooking a burger to a higher temperature will affect the juiciness as well due to evaporation. So the methylcellulose addition, the method of adding methylcellulose, again, a binder that's commonly used in plant-based meat. In many products and in a lot of sources in literature, methylcellulose is simply added as a powder, as a dry powder, and water, oil, and methylcellulose are added separately to the texturized vegetable protein or beef. In this case, we used a different approach where we pre-mixed water, oil, methylcellulose, and then added it to TVP or beef. So we made a pre-emulsion or a pre-foam, which is something I haven't actually come across much at all. So here I just want to briefly show this PCA plot again, but actually all I want to show is that we have a broad range of, if we look at the juiciness scale, a broad variety, and the plant-based burgers, again, are on one side and the beef burgers on the other side. But within the plant-based burgers and within the beef burgers, we were able to generate different levels of juiciness, which is important because we're trying to understand where juiciness comes from, how to measure juiciness, etc. So again, we have our two methods of including methylcellulose. And what we found from our sensory results, again, carried out with a trained panel, is that adding methylcellulose powder and methylcellulose foam to beef gave a 10-step difference in sensory scores. Sensory score usually range from 0 to 100. And even for TVP, we were able to increase from a score of 20 to a score of 26. And that's only initial juiciness, but the TVP foam samples also had a lower final dryness, and which tells us that overall the juiciness or the moistness lasts longer. So this method of adding methylcellulose is, a way to improve, an effective way to enhance juiciness. So we now know that sample formulation affects juiciness. And when I say sample formulation, I'm referring to the different oil and water content and the methylcellulose content and the preparation. Our next question is, so just to summarize again, we have beef burgers that are higher in juiciness, again, overcooking burgers, which I hadn't shown in detail, but as you can imagine, again, cooking for longer decreases the juiciness due to evaporation. A higher amount of methylcellulose, which I also showed for the tempeh burgers, decreases juiciness and increases firmness in TVP and in beef burgers. And adding methylcellulose as a premixed foam instead of as a dry powder increases juiciness. These were some of our main findings. And the next important question is, how can we accurately measure juiciness mechanically? Now, we were not the first one to try to tackle this challenge. Again, juiciness has been identified as a very important attribute in plant-based meat, but also in animal-based meat, but also in fruits and vegetables. So some existing methods include measuring water release in a system that's completely submerged in water. So this method was developed in 2020. Again, this is a very relatively recent, so looking into plant-based meat and the juiciness is relatively recent. So to me, a bit of a disadvantage or a challenge for this is that it's submerged in water, which becomes challenging if you are using a system, for example, that easily breaks apart in water, disintegrates in water, or that contains a lot of oil. I thought this was also an interesting approach. In this case, they carry out compression on a filter paper, and then they look at the stain that was left behind, and they use image analysis and machine learning to try to determine how much juice was released, which can tell you how juicy the sample is. Unfortunately, these authors did not use any sensory analysis, so it's difficult to know whether that image analysis method closely mimics sensory evaluation. And then a more recent method where a compression method was applied, and the sample was, or the chamber was connected to a vacuum pump, and the vacuum pump then assists in extracting, using pressure, extracting the liquid. So in our approach, we want to measure juiciness in a way that closely mimics sensory evaluation. So for this, we resorted to our texture analyzer, which I mentioned before, which is a machine that compresses samples and can measure the hardness. But we did not use it to measure hardness in this case. We actually used it to compress a sample, and we have filter papers attached, and we measure at a range of speeds and deformations, and we actually measure with the burger crust on and or off. And removing the crust is also something that had not been done before, and which for us actually yielded very interesting results. So what we do is compress the sample, and we have filter paper with some fluid that's been released from the burger, and we actually weigh the filter paper before and after to know how much fluid has been released. We use that compared to as a function of the total sample mass, and from that we analyze and calculate our compression loss. So in our case, the juiciness measurement, or the juiciness measured, the liquid released upon compression, we refer to as the compression loss. So I don't want to go into too much detail here, but I just want to show you what some of the raw data looks like. And what you can see here is the different measuring speeds, and we measure with the crust on and off. As expected, beef has a higher compression loss than TVP burgers. At 0.1 millimeter per second with the crust removed, we get the highest compression loss across all samples. The water and oil expelled, which I show in gray and blue here, depends on the sample type, but it also depends on the measuring conditions. And now the actual question we wanted answered in this project is, how does the compression loss relate to our sensory results? To have a better understanding of this, we carried out a Pearson correlation, and I'll show you the correlation diagram of the total compression loss. So what we've done here is correlating our measuring conditions, so the different speeds, with crust, without crust, to a range of sensory attributes, juiciness, oiliness, etc. And what we find is that for all attributes, measuring at 10 millimeters per second with the crust removed gives the strongest correlation. So that means this measuring condition is closest to sensory evaluation for both beef and TVP samples. And this is important because when carrying out a measurement like this and selecting the settings, or collecting the way you're going to measure your sample to relate to sensory analysis, that will ultimately determine whether the method is accurate enough or robust enough to involve in sensory research. And here we've shown that this method actually is quite robust across a number of sensory attributes. So why is the measurement, this faster measurement, so 10 millimeter per second, without crust, more accurate? So first of all, we're measuring on filter paper. That means that if the sample sits on the filter paper for longer, so a slower measurement speed, there's some absorption taking place, which means that we're not only measuring the fluid released during compression, but there's also some absorption by the filter paper. So when we measure a faster speed, this is why we think we get a stronger correlation. When measuring with crust, I'll just show that again. When measuring with crust, the fluids are mostly released through the sides. This is because the crust is quite hard and quite dry, and actually prevents fluids from being released. When we remove the crust and carry out the same measurement, fluids are allowed to be released from all directions, which we can imagine closely mimics what happens during mastication when you actually fully chew and masticate the sample, and fluids are released from all directions and from the different fracture sites on the sample as well. So that's why the 10 millimeter per second without crust gives strong correlations with mastication. So to summarize our findings on juiciness, as I mentioned several times, beef samples indeed do have higher juiciness than TVP samples. We were able to measure that as well for the compression loss. The addition of methylcellulose as a foam increases juiciness in TVP samples, and compression loss at 10 millimeter per second with the crust removed closely mimics sensory results. So this work was also recently published in Food Hydrocolloids, and while I have this slide up, I'd like to also thank the authors who contributed to this work. So to summarize, as we're reaching the end of my presentation, we've actually gone over a few important things. We identified, not necessarily in this order, but we were able to identify important sensory attributes for plant-based meat analogs. We developed novel methods to characterize these material properties, including juiciness. And we now also understand how different molecular interactions within fermented legumes can generate specific properties. So we know how to generate the properties, we know which ones are important to meat and plant-based meat analogs, and we know how to measure them. So again, this is what I showed you at the start. Probably the first time seeing this image, you might have felt a little bit confused or a little bit uncomfortable. I do encourage you, if you're at any international supermarket or international restaurant, I challenge you to try some of these products, and you might actually be surprised at how much you enjoy them. So where does the potential lie for food fermentation, in my opinion? So the legume fermentation process is a rapid and sustainable, or relatively sustainable if you compare it to some other production processes. We have tunable textures and flavors, which opens up a lot of possibilities, which means we might not only be able to make something that resembles meat, but beef, but maybe also fish or other meat products, or maybe even new flavors that aren't even related to any meat products we know. Maybe we can just make new flavors that are enjoyable and acceptable in their own right. There are post-fermentation opportunities and potentially novel plant-based foods with enhanced protein functionality, which means this might not only be limited to plant-based meat, but possibly also plant-based milk or other plant-based products that we can find on the market. So I'd like to acknowledge the professors, students who have contributed to this work, and of course, a big thanks to Heather, who's led this research over the past 3 years. This work was in collaboration with Motif Foodworks, and as was already mentioned, it was an ARC linkage project. And in the future, I will hopefully continue working on fermentation, and there will be possibly ancient fermentation still, but also more advanced approaches to fermentation, such as precision fermentation, as was also mentioned earlier. So I'd like to thank you all for your attention. And Heather, I'd like to switch back over to you for the Q&A session.
Fantastic. Thanks, Raisa. What an interesting presentation. You're such amazing presenter. It was a pleasure to listen to you. So please get those questions into the Q&A now, please, everyone, not the chat, into the Q&A. There's a couple there already. But I just wanted to ask you, Raisa, how much of those different fermented products have you tasted yourself, out of curiosity?
So I have had Tempeh. So Tempeh is the only one that I was quite familiar with before starting the project.
Yeah.
I have also had natto quite a fair bit, and that's actually a bit of a guilty pleasure. It's definitely an acquired taste, but if you have it with rice or egg and enough soy sauce, it's actually quite pleasant. You can maybe compare it to Vegemite. When you have it on its own, it's very intense, but then you have it with some toast and butter. It actually works together quite well. And we've recently experimented a little bit with natto in the lab as well.
Yeah, awesome. I imagine it's hard to get hold of some of those other ones.
It is, yes.
Yeah. As a flavour or a volatile flavour chemist, I'm really interested in the volatile notes of these. You mentioned some of them have some of those meaty flavours like sweetness or umami that are often derived from glutamate and other types of proteins that cause sweetness in protein products. What about the aroma notes, the volatile notes that are present? Are they meat-like as well?
Yes, some of them are. So I've actually removed that to keep it a bit more dense, but that's a very interesting question, Heather. So if we think about beans, especially cooked beans, they have a very distinct beany, legumy, maybe green herbal flavor. And during fermentation, a lot of those beany notes are actually reduced. A lot of those green, grassy notes are reduced. And the aromas are often described as roasted, maybe even popcorn-like for foods such as tempeh. And natto, it's a bit more of a blue cheese, nutty aroma. So yeah, the volatiles definitely are affected by fermentation as well.
Nice. They really are the original plant-based meat.
Yes.
Aren't they? Now, a question from someone who has participated today. Thanks for popping it in the Q&A. Thanks, Raisa, for a great presentation. Why do you using different binders change the flavour of tempeh differently?
That's a very good question, and possibly ties into the previous question a little bit. So I'll keep it simple. So I mentioned methyl cellulose, for example, as one of the binders that improves the firmness, but also reduces the moistness or juiciness. And the reason that happens in the case of methyl cellulose is because it actually binds water. Now, if we have some water-soluble flavors, and the water is entrapped by methyl cellulose, that means that those water-soluble flavors really aren't at the forefront of what we would taste because there's not as much water available. Now, for gluten has its own distinct taste, which might be contributing to a change in flavor as well, and the same for egg white. So, and also having the binders so it essentially contributes to how the water and oil is distributed in the system, and it's the flavor of the binders themselves. So I think it's those two. But we haven't actually carried out the measurements to know this, but I'm quite convinced that's what's happening.
For sure. And without them being available, they're not available to the taste buds on the tongue, or even to our olfactory receptors. They're tightly bound, and even chewing them doesn't release them.
Yeah.
Another question here, Raisa. Great talk. My question is about the fat composition of the fermented grains. You mentioned that there's a high content of stearic acid, which is related to fishy, grassy odours and correlates to negative consumer appeal in meat. Do you know if fermented grains retain a significant monounsaturated fatty acid content that can offset the saturated fat part of the aroma?
Yeah, good question. So I was able to identify 4 fatty acids. I only showed 2, but off the top of my head, there would have been oleic acid, linoleic acid, stearic acid, palmitic acid. And they are usually associated with a maybe a bit sour, acidic flavor, and not necessarily a quality component in meat, but that does depend on the percentage that's present. In most fermented foods, the percentage is quite low, but it does depend on how long the fermentation goes on for. And I believe that the longer they're fermented, the higher the percentage of those fatty acids become as well, which is why the longer fermentation often leads to a more intense and more off flavors as well.
Yes. Thanks, Raisa. Next question here. Great work, Raisa. During beef patty processing, do we add ingredients such as salt, which also affect juiciness? Since it was not considered in your study, is it not a significant factor?
So salt definitely is an important factor. Salt improves the protein solubility especially in beef burgers. That's quite known. I don't know if anyone's actually researched how it affects plant-based burgers in terms of solubility, especially because those proteins have poor solubility. But in the model system where we did use beef burgers, we intentionally did not use salt. We just kept everything as bland, as basic, as simple as possible. Because in that study, we actually did not ask our panelists any flavor questions either. We really fully wanted them to focus on juiciness, as our intention was to get juiciness results that are very reliable, very robust, so we can relate them to the measurement we were developing.
Awesome.
That
Thanks,
answers the
Raisa.
question.
I was going to ask you myself as well, in your reading, I know you wrote a review that's been published on these ancient fermentation techniques. Are there any, I guess, risks associated in terms of, fermentation's gone wrong, or species that might come in and cause toxic compounds? Did you come across any of that with some of these more unusual fermentation practices, or are they pretty robust where they're used?
So the answer is yes, there is a risk. However, as long as everything that is used is being sterilized, either using alcohol or by boiling everything for 3 minutes to get rid of any other bacteria, to get rid of any cross-contamination. We have had some bad batches, but especially with tempeh, it's something that's done at home.
Yes.
In restaurants, it's done in environments that are a lot less controlled than our lab environment and possibly even a lot less clean. So as long as everything is kept very clean, the risk is limited. And with tempeh in particular, you can tell from the smell, you can tell from the colors. And we haven't had any issues ourselves, but it is definitely a possibility.
For sure. So I think someone who's experienced at making it or who's been shown how to make it and what to look for might be able to know if things have gone wrong.
Exactly.
Another question, Q&A. A good presentation, Raisa. I know dawa dawa, the African fermented bean product, is very pungent. It will be interesting if it can be used in plant-based meat, but how do you mask the pungent smell? That will be a big task. Do you have anything thinking about this?
Yes, that is a very interesting question, and especially because I only know of this product on paper from literature. And the most interesting approach I've found to reduce that aroma is using radiation. So radiation approaches to affect those flavor and aroma molecules that cause those pungent flavors. I've also read that boiling for a longer time might reduce it a little bit as well. And I think that the fermentation time, reducing the fermentation time, would also reduce the intensity. However, when it's traditionally made, it's made for that intense flavor. So it's a bit of a paradox, a bit of a contradiction to reduce the fermentation time, to reduce the flavor when the flavor is what's the quality aspect in this case. But I think possibly maybe even combining dawa dawa with something like tempeh, which has less of a strong flavor, could be an approach, or using different microorganisms combined. I think those are different approaches that could be researched in the future.
And could be used as a minor ingredient because of a particular property. Also with volatile aroma compounds, depending on the concentration of them present, they can really range in terms of what those pungent smells might be. If it's at higher concentrations, you can have a very different smell. There's a compound that smells like cat's urine at high concentration. At medium concentration, it smells like blackcurrant and passion fruit. And then at low concentration, it has another note as well. So it's it certainly the concentration can change things. I'm not sure if you'll know the answer to this question, also from Samson. If we reduce the days of fermentation for dawa dawa, do you think we will get the same dawa dawa product? I guess he's just thinking about reducing that pungency.
Exactly. Yes. So that's what I just said. That's where a bit of the contradiction comes along. So reducing the fermentation time will reduce the pungency. I'm convinced that's the case, but it will also change the textural properties. Now, I have no idea what the textural properties will be like if the fermentation time is slightly reduced, but it could also be that the textural properties are something that more resemble what is consumer acceptable when thinking about beef products. But that is I wouldn't even call it a hypothesis. That's a wild guess, but definitely something interesting to think about for future research.
Awesome. Raisa, thank you. It's 1 o'clock. Really appreciate you giving up your time and preparing such a fantastic presentation. Thank you everyone for joining today. I really appreciate you coming along. I'll let you get back to your other bits and pieces. There's one more slide. If you haven't already subscribed to the Coffee Science Seminar Series, you can quickly get your phone and grab this QR code and sign up to receive emails about this seminar series. So thanks once again and enjoy the rest of your Tuesday.